Hello all, I am Diborah Gutema and this is my video presentation for my project's pharmacological characterization of chimeric NMDA receptors in Xenopus laevis oocytes. NMDA receptors are ion channels located on neurons that allow calcium ions to enter the cell when activated by the neurotransmitter glutamate. This calcium signaling, known as ionotropic signaling, is critical for synaptic plasticity, learning, and memory. NMDA receptors can also engage in non-ionotropic signaling, where conformational changes in the receptor trigger internal signaling pathways without ion movement. Each receptor is composed of 2 GLUN1 subunits and 2 GLUN2 subunits. A developmental shift occurs where GLUN2 subunits are gradually replaced by GLUN2A, a transition that is essential for synaptic maturation. Understanding how these subunits contribute to ion flow and conformational signaling is the key focus of our project. To investigate how different regions of NMDA receptor subunits contribute to signaling, we are working with chimeric GLUN2 constructs developed by Dr. Dumas's lab. These chimeras are engineered by swapping specific intracellular domains between the GLUN2A and GLUN2B subunits. In doing so, we can separate the functional contributions of individual regions, such as the intracellular tail, to ion flow and to non-ionotropic signaling. By studying receptors with these controlled domain swaps, we aim to determine which portions of the subunit structure are responsible for differences in calcium permeability, activation properties, and downstream signaling. This semester, we focused on preparing the DNA constructs necessary for expressing these receptors in Xenopus laevis oocytes for future functional testing. The overall goal of this project is to express wild-type and chimeric NMDA receptors in Xenopus laevis oocytes and compare the ionotropic signaling properties using 2 electrode voltage clamp recordings. By analyzing how domain swaps between GLUN2A and GLUN2B affect receptor function, we aim to better understand the molecular basis of NMDA receptor signaling. This semester, we focused on preparing high-quality plasmid DNA, optimizing restriction digest, and initiating PCR amplifications of the GLUN2 receptor inserts to prepare for future subcloning and expression studies. First, upon receiving the plasmid PGEM-HE membrane MEGFP, we transferred the sample from the backstab into a 3 ml bacterial culture, which was incubated overnight at 37 degrees Celsius for 16 to 24 hours. The plasmid includes a Xenopus laevis promoter sequence, which enables later expression in oocytes. Following incubation, we isolated and purified the plasmid DNA from the bacterial culture using an alkaline lysis mini-prep protocol. To ensure the integrity and purity of the plasmid, we assessed DNA quality using agarose gel electrophoresis to check for intact plasmid structure and spectrophotometry to measure the 260-280 absorbance ratio for purity. Next, we performed the restriction digest to prepare the plasmid for future subcloning. We used the enzyme NHE1 to linearize the plasmid and carried out diagnostic digest to prepare for the later excision of the GFP segment originally present in the vector. In parallel, we grew bacterial cultures containing the DNA for GLUN2A, GLUN2B, ABC, and BAC constructs. Using these templates, we initiated PCR amplification with construct-specific primers to selectively amplify the inserts. PCR amplification is currently ongoing. Once complete, we will purify the amplified products and verify insert size by gel electrophoresis before moving on to the next phase of subcloning. After the inserts are fully amplified and purified, we will digest them with restriction enzymes to create compatible ends with the plasmid vector. We will then use a DNA ligase enzyme to join the inserts and vector together, creating new plasmids that carry either the wild-type or chimeric NMDA receptor sequences. Some ligation reactions will be performed in-house, while others may be sent for commercial cloning, depending on efficiency. Sequence verification will follow to confirm successful ligation. Following sequence confirmation, we will synthesize capped RNA transcripts from the recombinant plasmids using in vitro transcription. These RNA molecules will then be injected into individual Xenopus laevis oocytes, allowing the cells to produce functional NMDA receptors for electrophysiological testing. 2 to 3 days after RNA injection, we will perform 2 electrode voltage clamp recordings, a technique that holds the membrane potential constant while measuring ionic currents. By applying glutamate and glycine, we will evaluate receptor function based on current amplitudes, activation and deactivation kinetics, and dose-response characteristics. Comparing wild-type and chimeric receptors will help us determine how specific subunit regions influence NMDA receptor ionotropic signaling. In conclusion, we focused this semester on growing bacterial cultures, isolating and purifying plasmid DNA, troubleshooting purification and digestion protocols, and beginning PCR amplification of the NMDA receptor inserts. These steps are critical for setting up RNA synthesis, oocyte injection, and functional testing. Moving forward, we aim to complete subcloning, synthesize RNA, and characterize receptor function using TEVC recordings. Finally, I'd like to take a moment to thank those who have been instrumental in this project. Dr. Teheran, who has been an invaluable mentor in electrophysiology and molecular biology. Dr. Dumas, who has provided expert guidance on receptor signaling and chimeric constructs. Hannah Zikria Hagemeyer, who was essential in training me on plasmid preparation. And finally, I'd like to thank the rest of the PB&J lab for their collective support through guidance and resources, which has been key to my growth as a researcher. Thank you all for your help and support.